Hello everyone. Today I would like to deal with the continuation part of physiology of digestion. Gastrointestinal hormones regulate the digestive secretion. Gastrin hormone is secreted by gastrin cells in the pyloric region of the stomach. It stimulates the gastric gland to secrete and release the gastric juice. Next hormone, enterogastrone. It is also called as gastric inhibitory peptide. It is secreted by the duodenal epithelium. It inhibits gastric secretion and motility. It slows gastric contraction. Hence, it is also called as gastric inhibitory peptide. Secretin hormone is secreted by the epithelium of duodenum and jejunum. It releases bicarbonates in the pancreatic juice. It increases secretion of bile and it decreases gastric secretion. Cholecystokinin hormone is secreted by the epithelium of entire small intestine. It stimulates the gallbladder to release bile and pancreas to secrete and release digestive enzymes in the pancreatic juice. Duocrinin hormone is secreted by the duodenal epithelium and stimulates the Brunner's glands to release mucus and enzyme into the intestinal juice. Enterocrinin hormone is secreted by the epithelium of entire small intestine. It stimulates the crypts of lubricant to release enzymes into the intestinal juice. Vasoactive intestinal peptide hormone is secreted by the epithelium of entire small intestine. It dilates peripheral blood vessels of the gut. It also inhibits gastric acid secretion. Willikinin hormone is also secreted by the epithelium of entire small intestine. It accelerates movement of villi. Small intestine is involved in the absorption of major part of the nutrients. The absorption may be through diffusion, osmosis, facilitated diffusion, and active transport. Absorption of carbohydrates. Here, all carbohydrates essentially are absorbed in the form of monosaccharides directly into the bloodstream. Blood carries these monosaccharides to the liver where they are converted to glycogen and stored in this form. Glucose and galactose are absorbed by active transport, means with the expenditure of ATP. Fructose and mannose are absorbed through facilitated diffusion, that is by the help of the carrier molecules. But again it is along the concentration gradient, means higher concentration to lower concentration. The diffusion is more rapid, but all the contents cannot be absorbed. Absorption of amino acids. Here, proteins are absorbed as amino acids directly into the bloodstream. Blood carries these amino acids to the tissues where they become part of the metabolic pool from which body proteins are synthesized. Amino acids are absorbed by active transport. Coupled with active sodium transport, the energy for the transport is supplied by a sodium co-transport mechanism. They also enter the bloodstream. The end products of fats are mixture of monoglycerides, fatty acids and glycerol. Glycerol is water soluble, so it is directly absorbed by the mucosal cells of intestine. The long chain fatty acids and monoglycerides are insoluble in water. In the intestinal lumen, fatty acids, monoglycerides, and the bile salts aggregate to form water-soluble molecules called mixed micelles, each being 0.03 to 4 nanometer to diameter. Each mixed micelle has the bile salts in the outer part, whereas the hydrophobic fatty acids and cholesterol. These molecules form the core of micelle. These micelles are taken by the intestinal mucosal cells, generally by the process of pinocytosis. Inside the mucosal cells, fatty acids and monoglycerides react to form triglycerides. The neutral fat so formed is surrounded by a coat of beta-lipoprotein to form water-soluble fine globules called chylomicrons, each about 1 millimicron in diameter. Chylomicrons are milky in color and are released in the limb capillaries called lacteals, from where these are carried into the thoracic duct and then into the systemic circulation. Absorption is the process by which nutrients produced after digestion are circulated throughout the body by blood and lymph and supplied to all the body cells according to their requirements. Practically all absorption takes place in the small intestine. The presence of villi on the inner surface of the small intestine and the presence of microvilli on the free surface of the intestinal epithelial cells enhances the absorptive capacity of the small intestine. Assimilation means the incorporation of the absorbed food materials into the tissue cells as their integral and homogeneous component. The various food molecules taken up by the cells are transformed as amino acids, monosaccharides, and fats. Thank you.